Hello everybody, and thank you for joining us today. I'm Dr. Martin Graf-Utzmann. I will be moderating this webinar today. Today's webinar is split in two parts. The first part is titled Highly Multiplexed Target Proteomics Acquisition on a TIMS-QTOF, and the second part is titled Leveraging prm-PASEF on the timsTOF Pro to Interrogate the Functional Proteome. Today's webinar is brought to you by Brooker. The first speaker, which I'm happy to introduce, is Professor Dr. Gunnar Dittmar from the Luxembourg Institute of Health. The second part will be presented by Dr. Jarrod A. Marto from the Harvard Medical School. Gunnar Dittmar is the head of the Proteomics and Cellular Signaling Group at the Luxembourg Institute of Health's Quantitative Biology Unit. He started his career with a Ph.D. at the German Cancer Research Center in Heidelberg, followed by 6 years at the Harvard Medical School in Boston, USA. He came back to Germany and was recruited at the Max Delbrück Center for Molecular Medicine in Berlin in 2003. 4 years later, he established the mass spectrometry core unit at the Max Delbrück Center, and in 2014, he additionally built up the proteomics unit at the Berlin Institute of Health, a translational research center resulting from the joint forces of the Max Delbrück Center and the Charité, the Medical School of the Freie and Humboldt University, Berlin. Since 2011, he is a visiting professor at the Technion University in Haifa, Israel. In 2016, he joined the Luxembourg Institute of Health, where he created the new proteomics research group, a bioinformatics group, and built the new multi-center LuxGen next generation sequencing platform, a joint effort of the LIH and the LNS, the Laboratoire National de Santé. In 2018, he started work on setting up the new quantitative biology unit, which was officially inaugurated in 2019. The unit consists of 10 different technology-oriented groups and platforms spanning from bioinformatics to in vivo imaging. Since 2019, he has been appointed as an affiliated professor for proteomics at the University of Luxembourg. Jarrod A. Marto received his Ph.D. in analytical chemistry at the Ohio State University with Alan Marshall and went to do postdoctoral studies at the University of Virginia with Don Hunt. After several years in the biotech sector, he joined the Dana-Farber Cancer Institute in 2004. Jarrod uses mass spectrometry as a primary discovery tool to understand how genomic alteration, environmental insights, or the action of small molecule chemical probes impact the functional proteome, including protein post-translational modifications, biochemical complexes, signaling pathways, or how they manifest or mask as phenotypic or disease signatures. His research is further enabled by development of advanced chromatographic separation platforms which provide improved peptide detection and quantification and support genome-scale interrogation of mammalian proteomes. In addition, his lab builds open-source computational tools that facilitate interrogation of mass spectrometry data across the full spectrum of scientific inquiry, from the underlying instrumentation to interpretation of quantitative proteomic data in the context of biological pathways. This research program represents a productive convergence of biology, computation, and analytical science. Before we get started with today's event, let me remind everybody that this is an interactive web seminar, so you can ask questions at any time during the presentations by clicking into the questions box. Only your presenters will see the questions, so don't be shy about asking them. Questions will be answered in the Q&A session at the end of the presentation as time permits. Now, if the speakers aren't able to get to your question today during the live broadcast, then they will do their best to follow up with you later offline. To answer one of the frequently asked questions before we start, yes, there will be certificates available for this webinar. Without further ado, let me turn the floor over to Gunnar, who will begin our webinar today. Okay, Gunnar, it's over to you.
So thank you for the organizers for giving me the opportunity today to present our research on the highly multiplex targeted proteomics acquisition, where we use timsTOF Pro to set up this new technology. So as probably most people know, proteomics analysis can be easily divided into several subfields. Probably the best well known is shotgun proteomics, and where we simply look at as many proteins as possible. A rather newcomer is data independent acquisition, where we can actually have something that is in between a targeted proteomic approach and a shotgun proteomic approach. And finally, we have targeted proteomics, where we are focusing on a very small set of proteins, which we can then measure with very high sensitivity. We can actually use very short gradients for this analysis. And we have the advantage over, for example, shotgun proteomics, that we can identify a set of targets in each experiment, which is extremely important if you want to look at patient cohorts or larger cohorts of any kind, to identify biomarkers and have a consistent quantification of these biomarkers in your cohort. So my lab is particularly interested in targeted proteomics, and we are using a special implementation of targeted proteomics called parallel reaction monitoring, or PRM. And since the timsTOF Pro provides very interesting new measurement features, we were setting out to implement this technology on the timsTOF Pro. So let's first take a look at the characteristics of targeted acquisition methods. Parallel reaction monitoring is implemented on high-resolution mass spectrometers, which have a certain setup. So we have a quadrupole, which we can actually use for the selection of the peptide. Then we have a collision cell, which allows us to break down the peptides into smaller fragments. And then we have a measurement unit, in this case, a time of flight, which allows us to record high-resolution spectra of these fragment peptides. And if we measure these fragments over time, we can actually integrate them and get a time-resolved profile out of that measurement. Integrating the area under that curve allows us to quantify the peptides with a very high accuracy. When we look at a TOF instrument, we can actually see that the TOF instrument has two types of resolutions built in. So the first quadrupole actually has unit resolution, while our TOF part of the instrument can actually record in very high resolution. And we can actually use this feature by following now the same peptide fragmented over time, and we will get several spectra of the same peptide with different features, where we can, for example, see here at first the fragment ions Y7, 8, and 9 are hardly visible, and then when the signal gets stronger, we can actually separate it very nicely from the background. To get an optimal result from this analysis, we actually have to know the retention time of the peptide that we are interested in, the charge, and knowing the charge, we can also do some optimizations on the collision energy, which is, in the case of the timsTOF, usually optional. So when we look at our three spectra, we can now place them in a timely order, and then we can actually see that we get a nice peak in the time resolved measurement. And we want to actually have at least 16 data points to have a smooth quantification here. Since peptides are not eluting all the time from a liquid chromatography system, but only in a very defined window, we only have to look for a specific peptide at a specific time in the chromatogram, and this allows us to actually focus our intention on specific peptides at a certain retention time within our chromatography system. When we now move from a normal TOF instrument to the timsTOF Pro, we realize that there's a very special front end at this instrument. It contains this second generation dual TIMS analyzer. And this is a ion mobility device that can actually separate ions by charge and shape. And this TIMS analyzer actually falls into two parts of this analyzer. And this allows us to use this ion mobility trap first as an accumulate and trap part, and then to elute all the ions that we are interested in at a certain time. So here we can actually see the dual TIMS analyzer, and we can actually use the first part of the analyzer to store the ions, and they are essentially as a steady flow in this analyzer are stored, while in the second part of the analyzer we can now separate them and measure them one by one when we release them from the second analyzer. And when we do that by lowering the energy, we actually get three different signals for our three analyses that we are separating here. So why is that actually interesting? And the first fact that is super interesting for targeted proteomics is that we can actually use this to increase our signal. So while we can accumulate for 50 to 100 milliseconds in the first part of the TIMS analyzer, the release can take place in a very short time of about 2 to 5 milliseconds, which increases our signal by almost 50-fold. So when we look at the ion flux of with the TIMS off, then essentially we can't get a signal, but with the enhancement of the TIMS on, we can actually squeeze our ions together and get enough signal so that we can still detect it. And this feature can actually help us to gain more sensitivity. So when we set up this method, we first set up a benchmark system. So we used 214 Aqua happy peptides. So these are peptides which contain an isotope labeled amino acid, so that we can actually separate it from the naturally occurring peptide. We generated some dilution curves for these 214 Aqua peptides, and then used some A509 human carcinoma cell line extracts. We digested them down to peptides, spiked in our dilution series of the Aqua peptides, measured the whole thing on the timsTOF Pro, connected to a nanoelute system, and then analyzed these mixtures first by TIMS-DDA or PASEF, and in a second system we analyzed it then by TIMS-PRM. And as a informatic pipeline, we used Mascot or Proteinscape, and for the analysis of the targeted proteomics, we use Skyline PRM. So when we now look at the effect of the TIMS ion trapping, the concentration effect. So when we look first in the upper row, we can actually see 125 attomole of a selected peptide. When we turn off the TIMS and only analyze it by the TOF part in PRM mode, there's hardly any signal, while when we are turning on the ion trapping, we can get a very nice shaped signal from the three fragment ions that we are following for this one. And if we are going one dilution step down to 31 attomoles, you can see in the TOF only analysis we can't see anything. It's just noise, while we still get a not very nicely shaped but still detectable signal in the TIMS-PRM with ion trapping. Now look at the limits of quantification. So what is actually the recovery of the signal that we get? And we can see that even in 50 attomoles injected, the TIMS-PRM almost detects everything that we injected, and only decreases very slowly to about 50% recovery with 31 attomoles injected, while the same instrument run in TIMS-DDA or PASEF mode loses sensitivity much faster than in comparison to the TIMS-PRM. When we now move to a multiplexed detection using the TIMS-PRM, we actually faced a technical problem, and this problem is actually the coordination of the ion selection in the first quadrupole of the instrument with the ion mobility. So when we actually want to isolate a peptide that is separated by ion mobility, we have to coordinate the Q1 filtering with the ion mobility. So when we use the TIMS-PRM and use a separation in the ion mobility of 1 target per 100 milliseconds, we can actually remove noise very efficiently from this analysis. When we move now to a multiplexed analysis, we have to coordinate the Q1 filtering with the ion mobility filtering, and so that the overlap of the two selection windows is coordinated, and this gives us a way that we can actually jump from one frame to the next one all across the selected peptides. This allows us to actually isolate several ions at the same time, which then are consecutively eluted and analyzed by the QTOF. So when we look at the IMS filtering, this allows us to separate interfering ions from the ion of interest. And you can see in the upper spectra, we have some ion mobility traces here. The upper trace is the peptide of interest for us, while the lower trace is something that is interfering. And when we measure that and integrate it over time, then you can actually see the separation here with a significant shoulder in the illusion profile. So when we now apply ion mobility filtering, filtering everything out that is grayed out and just focusing on the light part, which are corresponding to the shoulder part and the actual ion of interest, we can see that now the filtered ion of interest has a very nice signal to noise, almost no interference of anything else. So this actually gives us a very good way to quantify a peptide. And if you think of the combination of liquid chromatography, quadruple 1, MS2, and IMS, so we have actually 4 dimensions of selectivity that are now used in this analysis. So this is now an example for a multiplexed analysis, and each of the boxes in the upper graph represents one of the peptides that are selected. Yeah, so we have the ion mobility windows here, and so what actually happens if 2 of these windows are actually overlapping? Yeah, so we have here indicated with this little red line 2 frames that are overlapping, and so now our software actually is clever enough to select 2 different ions in the first round, ignoring the red one on the very right, and only selects it in the next frame in the next 100 milliseconds for the next acquisition. So developing this was actually the main challenge in bringing PRM to the TIMS-QTOF Pro. As an example for application of the TIMS PRM method on a real life sample, we used the well-described oncogene KRAS. And KRAS is frequently mutated in the position 12, and this mutation can actually mutate the glycine to a number of different amino acids, with the amino acid exchange G12V as the most frequent one. The N-terminus of KRAS is identical to 2 other proteins, NRAS and HRAS, and so we can't really distinguish these proteins by just looking at one of the peptides. So we used specific peptides for the G12 mutations. So we have isotope labeled peptides that are describing this mutation. And then we have 3 other peptides which are located in the C-terminus of KRAS, NRAS, and HRAS, which are surrogates for the quantifications of these 3 proteins. So first we quantified the 3 different RAS variants, KRAS, NRAS, and HRAS, in different cancer cell lines. So the first 6 cell lines you can, which are depicted here, are all lung cancer cell lines, followed by 2 colon carcinoma cell lines, and then we also got access to patient-derived colon cancer cell lines. And what you can easily see here is that KRAS, NRAS, and HRAS are significantly differently regulated depending on the cell line you have here. And with KRAS and NRAS being expressed at similar levels, while HRAS is in 3 cell lines only very lowly expressed, so that it's hardly detectable even by targeted proteome analysis. After measuring that, we moved on and to show that we can actually identify single amino acid exchanges. And here we used again our peptides with the different amino acids. And so we have first the total amount of KRAS that is measured. And then we can actually detect mutations in several of these cell lines. So, for example, the SW480 and 620 cell lines, which are colon carcinoma cell lines, carry the G12V variant. So when we look at the G12S mutation that is detected in the A509 cell line, so it's very specifically recognized here, and all the other cell lines which are genetically characterized clearly show no signal. So we have a very specific possibility to actually probe for single amino acid exchanges in these different KRAS mutants. Another example for the high specificity of the measurement is that the G12D, G12A, and G12C mutants are not detectable, which is in line with the genetic data. So when we actually moved now from back to our complex mixtures of peptides, we also analyzed how many peptides we can actually multiplex in a cell extract. And so we analyzed the 214 synthetic peptides in a 20-minute gradient in cell extracts. And what you can actually see here is the linearity of the signal. In the upper part, it's just the area signal, and you can see there's a slight drift in the upper and the lower part. But if we are using our isotope labeled internal standard, this is a perfectly linear, perfect line, and it's very linear. So we get very confident quantification over several orders of magnitude. To show that this is not only true for the analysis of peptides in cell extracts, but also in human serum, we took 260 peptides now, spiked them into human serum, analyzed that on a 20-minute gradient, and again you can see that the signal is very linear over a large portion, several orders of magnitude. And it's even better if we are putting in our isotope labeled peptides as an internal control, so that we have a very robust quantification in these samples. So in summary, we developed a targeted acquisition method with the timsTOF Pro. It allows us to analyze samples with enhanced sensitivity by ion concentration, so trapping for 100 milliseconds, releasing for 2 milliseconds, removal of interfering signals by Ims filtering. We can actually use significant multiplexing, and we use this for the analysis of KRAS, NRAS, and HRAS in cell lysates, and we quantified the mutations of the different RAS version in position G12 in these complex samples. Finally, I want to thank the people who were involved. So Antoine Lisieux and François Bernardin from the LIH working on the implementation of the prm-PASEF on our side. Many thanks to our colleagues from Brooker, especially Pierre-Olivier Schmidt, and then Sven Bremer and Jens Decker from the informatics side. And many thanks to our collaborator from the University of Luxembourg, Elisabeth Letelier, for sharing some biological materials that we can actually analyze in our systems. Thank you.
Thanks, Gary, for the kind introduction and invitation. And thanks, everyone, for tuning in to the webinar today. Happy to present our effort to develop advanced PRM acquisition schemes on the Brooker timsTOF Pro instrument. Before I get started, I do need to make these disclosures, and I'll note that near the end of the talk, I'll show a little bit of data from a 908 zip chip interface to the timsTOF Pro. Original development effort on PRM on the timsTOF focused on 68 candidate proteins that we had quantified across a Parkinson's case control cohort of CSF samples. So for our validation cohort, we turned to PRM on the timsTOF Pro, and we're really pleased to see across replicate injections of samples in our validation cohort, we could get great linear response, good reproducibility for peptides at high abundance as well as those at lower abundance. And you see that in each case, we got really great, rich MS-MS spectra, really supporting robust quantification by PRM. And here you see a few examples from a larger data set of proteins measured by PRM in our validation cohort. You see many measurements which agree nicely with the original data in our discovery cohort. So over the course of our development efforts with PRM, we were able to observe really nice reproducibility for large numbers of injections of CSF across more than a month. So it's all very encouraging. But of course, these methods are not perfect. So ideally, you would have a fixed PRM targeting window where you would have your peptide eluting right in the center of the targeting window across many injections across different samples. But of course, what we see in real life, especially with nanoflow liquid chromatography, is that our analytes will experience drift over the course of many injections, sometimes pretty significantly, such that the elution profile starts to sort of walk out of the targeting window, which of course will have a pretty negative impact on your ability to achieve reproducible quantification by PRM. And below, just a couple snapshots of this type of drift that we see for several peptides in our original validation efforts. So these limitations motivated us to expand our work with Brooker to implement what we call prm-Live. The idea is to use a combination of reference peptides and some code implemented through the instrument API to achieve real-time adjustment of the PRM window so that we can maximize the number of peptides acquired in a single run while maintaining robust quantification. And I'll show you a couple applications of this technology in selectivity profiling of small molecule enzyme inhibitors. So this slide illustrates our initial setup and testing of prm-Live. We began with tryptic peptides from HeLa cells, and we acquired passive DDA data using 45-minute LC gradient as a reference. For 80 peptide targets out of the digest plus a set of reference peptides, we record M/Z, mobility, and retention time parameters. Then we set up the prm-Live acquisition scheme and again use 45-minute LC gradient, rerun the same mixture as a validation. And then to test, we use a shorter 30-minute LC gradient, again injecting the same mixture, or a longer, in this case 60-minute LC gradient, again with the same mixture, and use these as test cases to confirm that prm-Live can dynamically adjust the targeting window to capture the same set of 80 peptide targets. This slide shows some data from our initial development effort. In the panel on the left, you see one peptide out of our set of 80 peptide targets. And here at the reference, the 45-minute gradient, the elution profile is nicely centered within the PRM targeting window. But when we shorten the gradient or make it longer up to 60 minutes, you see that prm-Live dynamically adjusts that elution window and accurately tracks this particular peptide. And in fact, the shift in retention is between 8 and 10 minutes relative to the reference. A pretty extreme case, but nicely demonstrating the ability of prm-Live to track these peptides. Next, we can look at the entire set of 80 peptide targets. And again, we see that the measurements returned by prm-Live very nicely correlate with the reference at the 45-minute and then the shorter and the longer gradients as well. Again, nicely illustrating that prm-Live is able to dynamically adjust the targeting window for a large number of peptides, even under these sort of extreme changes in gradient length. So next we wanted to establish the maximum number of peptides that we could track in a complex proteome. So here we're targeting a little over 2,000 peptides from a tryptic digest of HeLa cell lysate. We're going to do this across a 60-minute LC gradient. And the cartoon legend here shows how prm-Live is going to attempt to maintain the elution profile within the early and late time points of the targeting window. So you can see here how it's dynamically adjusting the window throughout the entire length of the 60-minute LC gradient in an effort to maintain that elution profile nicely within the targeting window. So at the end, we're able to, from the original set of 2,014 targets, we see that prm-Live can maintain essentially the 100% of the elution peak for 1,875 peptides within that targeting window, and that's reproducibly done across 5 replicate injections. And impressively, fully 1,753 of these peptides are recorded across these replicates with CVs of less than or equal to 20%, as shown here. Again, really nicely illustrating how PRM Live is able to dynamically adjust the targeting window across the 60-minute gradient to track a large number of peptides in this complex proteome. Data on this slide illustrate how PRM Live can maintain the number of robustly quantified peptides across replicate injections for different amounts of sample injected or different widths of targeting window. But you see here when you turn off the PRM Live feature, the number of robustly quantified peptides drops dramatically. The box plots here illustrate that we're able to maintain a nice number of data points across the peak to help ensure robust quantification. So at the end, we see that the PRM Live acquisition framework maximizes the number of peptide targets while maintaining number of data points to ensure reproducible quantification for these peptides in the complex proteome background. One focus of our lab is to understand the intersection of small molecule inhibitors with the functional proteome. So we've set up a number of chemoproteomic platforms to be able to interrogate protein ligand binding at the level of purified protein across a target class, kinases for example, or look for on and off targets on a proteome-wide scale. For the next few minutes, I'll talk about our efforts to integrate PRM Live with our activity-based protein profiling approach using the timsTOF Pro. I'll first describe our use of PRM Live for kinase inhibitor profiling. This experiment is set up as an activity-based protein profiling assay where we take cells or cell lysates, we incubate with either vehicle or a selective kinase inhibitor. These mixtures are then passed across multiplexed inhibitor beads. These are beads that have a set of somewhat promiscuous kinase inhibitors immobilized on their surface. So it's essentially a competitive binding assay in which kinases that are pre-bound by the selective inhibitor are not really available to be enriched on the MIBs, and therefore they're represented at lower abundance in the PRM Live assay. At the end, we have a matrix of inhibitors bound to their kinases. Similar to our initial development efforts, we begin with analysis of cell lysates in passive DDA mode across a 90-minute LC gradient, build up a library of sorts of kinase peptides. We validate these using PRM Live on a shorter 60-minute LC gradient and look for the subset of reproducibly detected peptides that map to unique kinases. And we end up, for our final assay, with a set of 1,200 peptides spanning 220 kinases, which we can reproducibly detect across 5 replicate injections. Here are results for PRM Live kinome profiling for a small panel comprising both reversible and covalent small molecule kinase inhibitors. Each compound is incubated at a single point concentration. THC 531, for example, is a covalent inhibitor known to bind CDK12 and CDK13. And you see data from PRM Live nicely highlights CDK12 and 13 as being selectively bound relative to other kinases monitored. We first identified this compound, JZ128, as a covalent inhibitor that targets the understudied kinase PKN3. Here we perform selectivity profiling by PRM Live to identify other kinases that are targeted by JZ128. And here we use prm-live across a concentration range in order to establish dose-dependent binding of JZ128 to the kinase. And encouragingly, we see good agreement between prm-live binding data and target engagement as established by Western blot. As another example, I'll describe our effort to use prm-live to profile the binding behavior of compounds that target deubiquitinating enzymes. Through the process of deubiquitination, DUBs will stabilize the expression of their substrates, shielding them from proteasomal degradation. The DUBs are considered an emergent class of drug targets in multiple human pathologies, including functional links between specific DUBs and oncoproteins that drive various forms of cancer. Inhibition of specific DUBs might then drive selective degradation of oncoproteins, providing a novel approach for cancer therapy. We have access to a first-in-class library of covalent inhibitors against this family of 100 DUB enzymes, courtesy of our colleague Sarah Burledge at the Dana-Farber. Here again, we'll combine prm-live with activity-based protein profiling. We have an activity probe that consists of an electrophile which targets the catalytic cysteine in the enzyme, followed by ubiquitin, which mediates selective binding of the target class of DUBs, and finally a biotin handle to enable biochemical enrichment. So DUBs that are bound by one of our covalent inhibitors are not available for binding by the ABP, while those that are not bound by a specific inhibitor are strongly enriched by use of the activity-based probe. This schematic shows how we set up our activity-based protein profiling experiment. We incubate cells with vehicle, or one of 4 covalent DUB targeting compounds, co-incubate with the DUB activity probe, followed by enrichment using the biotin affinity handle, trypsin digest, peptide cleanup, and then we encode peptides from each condition using TMT 6-plex isotope reagents, and go to one of 2 separation mechanisms on the timsTOF Pro. We first used standard LC chromatography, but then to explore whether we could increase throughput, we also analyzed these samples on the 908 device's zip chip electrophoretic separation system, here shown integrated directly with the timsTOF Pro. Here we're comparing passive DDA data acquired on each separation platform. We have spectra for the tryptic peptide from USP10 on either the LC or the zip chip platform, and you see very high quality MS-MS spectra generating high confidence peptide identification scores. As described earlier, we use the passive DDA data to build up a set of peptide targets. We then switch into prm-live mode on the timsTOF, and you can see even on the zip chip separation platform, where peak widths tend to be only a few seconds wide, thanks to the acquisition speed on the timsTOF Pro, we get very high quality MS-MS spectra, and at the same time maintain a good number of data points across each elution profile. And here you see the combined strength of prm-live on the timsTOF Pro with the ballistic separation afforded by the zip chip platform. We're successfully targeting 50 peptides spanning 33 DUBs in only a 3-minute elution window. We really are approaching warp speed in our ability to profile these DUB inhibitors. We also assessed quantification. Here you can see a comparison in PASEF DDA mode between LC and zip chip separation formats. And you see that relative to some references, we're able to maintain good quantification accuracy. And similarly, when we switch to PRM live mode and drive at warp speed, here again, you can see we're able to maintain accurate quantification in our analyses. Here we compare DUB inhibitor binding data between the 90-minute standard LC PASEF DDA acquisition and our 15-minute ballistic acquisition using PRM Live and the zip chip timsTOF combination. In this case, we compared binding for 4 different covalent compounds from our first-in-class library of DUB targeting molecules. You see that we get very good agreement for the specific DUBs bound by each inhibitor, as well as the extent of competitive binding recorded for each of the small molecule inhibitors by each approach. We have a bit more work to do to increase the landscape of DUBs that we can monitor in human cell lysates, but we're well on our way to establishing warp speed activity-based protein profiling for these DUB inhibitors. So in summary, PRM Live can significantly increase the number of peptides available for targeted MS-MS. Our colleagues at Brooker can address this point in more detail, but we found it to be a very open framework, which I think will really empower scientists pursuing a wide diversity of research projects. And in today's talk, I hope I've shown that PRM Live is a really powerful approach for activity-based profiling of small molecule inhibitors. And finally, the best part, no cleaning, no failed turbopumps, just data. Finally, I want to make sure to acknowledge everyone who played a role in all of the results that I presented today. In particular, in my laboratory, Eric Zhao, Scott Vaccaro, and Adrian Chan for all the work in developing PRM Live and the activity-based protein profiling. In addition, we're fortunate to have a number of great collaborators that helped us with all the projects described today. I also want to thank the whole crew at Brooker. They've been great collaborators. They really put their back into it and invest a lot of calories to help ensure our success. And finally, thanks to all of you for listening today.
Okay, many thanks, Jarrod, for your presentation to us today. So now we have a little time for our question and answer session. If you haven't yet submitted a question and you would like to do so, then now would be the perfect time to do that. So let's get ahead and see what questions have come in so far. Okay, so the first question is the quantification of the KRAS mutation is interesting. In your point of view, what is the advantage of measurement of protein mutation versus DNA or mRNA mutation, since genomics or transcriptomics can do much better than proteomics?
Okay, so can you hear me actually?
Yeah.
Yeah, that question is definitely for you.
Sorry about it. Okay, so why measure the mutations by proteomics? Actually, when you look at expression profiles of different cell lines for something like KRAS, you see actually a big difference between the transcriptome, how strong a gene is transcribed, versus what you actually see as a protein. So when you look now for mutations, and usually you want to do it for some kind of a diagnostic. You want to know if this mutated protein is actually there in a protein, it's really expressed. And our data actually shows that there's a big difference between the transcriptomic data and the proteomic data. So we see that for some patients, a mutation is actually stabilized and expressed on the proteomic level, even though that the mRNA is only very lowly transcribed. While for others, where you have gene amplifications, even for the mutation, the downregulation on protein level actually eliminates more or less the entire mutant, and these patients need to be treated in a completely different way.
Okay, thank you very much for this answer. Next question is, what is the maximum number of kinases you can monitor by prm-Live on the timsTOF Pro?
Hi, Jarrod here. I think that question is for me, I guess.
Yeah, I think so.
Sure. So I think the maximum, we can probably get up to about 275. That's a little dependent on cell line, of course, that you're doing the test in. And also in the data we showed here, we sought to kind of achieve a compromise to keep the acquisition at 1 hour or less. If you give up a little bit on the throughput, and you can extend the gradient acquisition time, and you can bump up the number of kinases in that way. So it's a little kind of a dynamic approach, I guess you would say.
Okay, thank you. I guess the next one is also right up your alley. Is the timsTOF Pro zip chip combo a good fit for proteomics applications?
Oh yeah, absolutely. We've been really excited about the data we're getting when we pair the really fast separation on the zip chip with the acquisition rate on the timsTOF Pro. It's a great combination. So we're really looking forward to pushing sort of additional boundaries and applications in the proteomic space with that combination.
Okay, thank you. Gunnar, I think this one is in your field of expertise. When do you need to use Aqua peptides?
Well, so in our data, like I showed in the last 2 slides, it's actually necessary to use at least crude peptides, not necessarily Aqua peptides, to normalize the signal in the very low part or in the very saturated part of the measurement to get a nice linear response. So then these heavy peptides are actually helping.
Okay. Thank you. So next question is the prm-Live. Is that a heavy reference triggering collection of endogenous peptide data?
Sorry, could you repeat? I didn't quite catch the second part of that. Could you repeat that, please?
Is it a heavy reference triggering collection of endogenous peptide data?
Oh, I think I understand. Okay.
Endogenous, yeah. Sorry, Jarrod, definitely for you.
Right. So in the way we've implemented that in the first round, we're basically triggering we're having the system dynamically monitor the IRT reference peptides. But I think it's a pretty flexible framework. So you certainly could envision using isotopically encoded heavy peptides as the triggering mechanism.
Okay, thank you. This is a rather broad question, and it's what is special about Brooker instruments, or about this Brooker instrument, I guess.
Well, I don't think it's meant to be that broad. What's special about it with respect to doing PRM? So maybe each of you could comment on that.
So I guess it's for me, then the question?
Yeah, of course.
Yeah,
You can.
okay, sorry. Yeah, what is so special? So the combination of the fast TOF in the back in high resolution with the the two-part ion mobility allows us to actually enhance the signal quite a bit, because the first part of the ion mobility we can actually use to store ions and then release them in a very short time, and that gives us a very high, very sharp peak that is then sent into the back part of the mass spectrometer to the TOF part, and that enhances our sensitivity quite a bit. So this is a wonderful combination of for the enhancement of the signal.
Yeah, and maybe if it's okay, I could just add, it's exactly the same comment, but just with different words. I think the integration of the TIMS separation is really sort of transparent, if you will, to the user, and it really does add this extra sort of separation, as Gunnar described nicely in his talk. And I think that really is helpful for the multiplexing. And then the other more general comment I'll make, in our experience with the instrument, there's no downtime. The thing just runs. We don't clean it. We don't have to change turbo pumps. There's no bullshit. It just cranks out the data, and that's enormously valuable.
Yeah,
yes,
I can imagine that. Sorry, Gunnar, I cut
you. Yeah, no,
short.
I can just say exactly, Jarrod, you nailed it down. It's a super reliable instrument. It's fantastic.
Okay, so the next question is, when will the PRM Live be available to the community?
Well, I think we're working very hard with Gary to kind of and the other folks at Brooker to kind of wrap up a couple loose ends, I guess you would say, and get that out to everyone in the community. I think Gary, obviously Gary would have to comment on real timelines, but I would just say that I think people are going to really dig it, and kudos to Brooker for being, I would say, very transparent in how they approach customer access into the system. I think it's going to be easy for people to adopt, and at the same time flexible to allow different types of workflows and creativity. So I think it'll be awesome.
Yeah, I hope it's going to make a big impact in the community. So how do you select the peptides to be targeted?
Well, so first we of course look for the best ionizing peptides that we can produce from a protein of interest, and then we test those peptides so that they are not interfering with each other in the ion mobility, so that we get a nice separation there. And not all peptides are behaving in the same way there. So you start with a couple of peptides, and then you select the ones which are performing the best.
Okay, thank you. So, Jarrod, this is one question for you. In your ABPP DAP screen, you see do you see dynamic range or chromatography problems caused by high abundant avidine or ubiquitin probe peptides?
Let's see. I don't think in that case that's not really been an issue that I can that I can see. We did choose to, in our first attempt, do the dub activity profiling by the zip chip because, there's roughly 100 of those enzymes in human cells as opposed to, 500 or so kinases. So we thought, as a first attempt, that the zip chip approach or the zip chip combo with the timsTOF, we thought the dubs would be a little bit more tractable, if you will, just based on the more modest size of the target class or the enzyme family.
Okay, thank you. Next question is the PRM Live available for all timsTOF users? Probably the question should be, will it be available?
As far as I'm concerned, I think that would be great. And I can't speak for Brooker, but I believe that is their intention.
Okay, thank you. The next question is, I'm working with cancer inhibitors, organic molecules. So how sensitive is the PRM Live for binding affinities compared to other methods?
Other methods, meaning other biochemical methods? Sorry.
I'm not quite sure. This is all I have in the question.
Oh, that's right. Yeah, sorry, it's not your question. Yeah, sorry. I think the one thing you have to keep in mind for this kind of assay, and it's completely independent of the fact that it's done on the timsTOF versus any other mass spectrometer. It's a binding. It's formally reading out binding. It's not. It's not really telling you enzymatic inhibition. Although often those two go hand in hand, but not all the time. So you do have to be a little mindful. Perhaps it's not so much about the sensitivity of the assay, but rather that the assay is reading out slightly different biophysical parameters compared to, a standard kinase assay, for example.
Yeah, I understand. So I'm afraid we are running out of time, but as I said earlier, the speakers will do their best to follow up via email with all the questions we couldn't fit into this session, unfortunately. What remains to say is that I would like to thank everyone for attending today's webinar. Thanks also to Brooker for all their support with this event, and of course a special thanks to you, Gunnar and Jarrod, for your presentation to all of us today. As a reminder, the webinar has been recorded and will be available for viewing in a day or so. And we'll send an email with the details on how to access all of this on demand. We'll send an email on how to access this on-demand version. So this concludes today's webinar. So from Gunnar, Jarrod, and me, it's goodbye.
Thank you. Bye.
Thank you. Bye-bye.